Hi, thank you. Yeah, so my name is Tychele Turner, and I just wanted to first start off by saying that I hope that everyone is doing okay during this current COVID-19 pandemic. I want to thank the organizers for the opportunity to speak today about sex bias and the genetics of autism. So this has been an area of considerable interest for me during my research career over the years. So the outline of my talk is as follows. I'm first going to tell you a little bit of the background of autism epidemiology and genetics and what we know about that today. I'm then going to tell you about the sex bias that occurs in autism and also in other disorders in the Diagnostic and Statistical Manual of Mental Disorders called the DSM-5. I'll tell you some potential biological reasons why this sex bias could occur. I'll show you some evidence for the female protective effect in autism, and then I'll spend the bulk of the time talking about our recently published study of de novo variants where we studied them by sex. And then we'll talk about how that feeds into gene regulatory networks more broadly in autism. So first of all, autism is a childhood neurodevelopmental disorder. It has a very high heritability, which is around 80%. It is fairly common in the population with a prevalence of 1 in 59, and it has a sex ratio of 4 to 1. And so you've probably heard this before. Basically what it means is that about 80% of all individuals with autism are male. Now, we collectively as a field have worked very hard to try to understand genetic and genomic factors involved in autism, and we have made a lot of progress. I would say that our first real big progress occurred in the early 90s, and this was when people were looking at single gene syndromic forms. So one that you may be familiar with is called Fragile X syndrome, and this is due to variation in the FMR1 gene. You may have also heard of something called Rett syndrome due to variation in MECP2. I just want to stop for 1 second and talk about FMR1 because I'm going to come back a little bit later and tell you how it kind of contrasts with some of the new findings that we have. So it's what we're finding is sort of complementary to this currently. But FMR1 is involved in Fragile X syndrome, as I mentioned, and how this occurs is there's an expansion of a repeat sequence on the X chromosome, and this primarily affects boys. Another area of investigation has been common variation, and I can say that at this point we're really confident that this type of variation is very important in autism. And we've done studies like genome-wide association studies and now more getting into polygenic risk scores. And we can say that their overall contribution is up to 50%. But right now we have about 5 genome-wide significant loci. And there's a lot more work that we have to do in this area to understand how it's really playing into the biology of autism. I would say that at this point in time, our greatest discovery in understanding of the biology of autism has come from our study of rare variation. And this rare variation includes things like copy number variants, which you may have heard of. And this is where you have either a loss or a gain of a piece of DNA. And the other are de novo gene disrupting mutations. And de novo means that the event is seen in the child, but not in the parents. So probably some of the most well-known copy number variants are 15q11 to 13 maternal duplications, as well as 16p11.2 deletions and duplications. Now there are actually many others, and most of them are recurrent. The other area that has really gained traction is in whole exome sequencing to find these de novo gene disrupting mutations. And as of our current estimates, the best list that we have right now is that we have about 124 genes that we can confidently say are involved in autism and other neurodevelopmental disorders from this approach. And why this is important is because we can begin to look at genotype-phenotype correlations once we know the genes. So for example, one study that I was involved in with our clinical collaborators was a study of a gene called DYRK1A. And this is a plot from one of our papers in 2017. And what it shows is different phenotypes. So intellectual disability or global developmental delay, speech delay, motor difficulties, microcephaly, feeding difficulties, and vision abnormalities, and then any combination of the 5. And what you're seeing in blue are individuals who have a variant in DYRK1A, and light blue are individuals who have a variant in DYRK1A that were originally ascertained because they had autism. And then we have in yellow the autism where we don't actually know the genetic variation that could be important. And we've subsetted that to the whole set and then the individuals who have intellectual disability. And what was really interesting to us was that when we looked at individuals with variants in DYRK1A, they often shared 5 or more of these different phenotypes. And so in particular, there are some where it's quite obvious that most people have that, like speech delay, and there are a few that are a little more variable, like vision abnormalities. But this is really critical as we move forward in trying to best understand autism in the context of the individual. So I'm now going to switch over to talking about the sex bias itself. And the first thing that I really want to convey to you is that while it seems somewhat mysterious, it's actually something that happens in many disorders. And I thought that I would just show you in terms of the DSM-5 in this plot, but I also wanted to point you to this review by Ober et al. In 2008, where they actually talk about sex bias more broadly, including outside of the DSM-5. But what I'm showing you here in this particular graph is on the x-axis, we have different disorders within the DSM-5. And on the y-axis are the fraction of cases that are male in that particular phenotype. And so on the left-hand side, you can see those which are biased towards females. And on the right-hand side, you can see those which are biased towards males. And so if we just took a look at one of them, anorexia nervosa, we can see that only 9% of all cases of anorexia nervosa are male. And if we contrast that with intellectual disability, where 57% are male, or autism, where 80% are male, we can see that there are quite a bit of differences when we look at these different phenotypes. So I think that because we see these features, it means that it's important that we consider it when we're studying these phenotypes. And it's important that we consider it on a genetic level as well as on a non-genetic level. So hopefully I've conveyed to you that these biases are not exclusive to autism, but do occur in other disorders as well. So why could this occur? And there are potential reasons that we've been able to come up with over the years. And I would say that the very heart of it is rooted in this thing called the liability distribution. The liability distribution was really first brought online in the 1960s. And it's a way that we can think about different phenotypes where we know there are genetic components as well as environmental components, which is an important consideration. So if we look at this distribution, it's not complicated to understand actually. We have on this axis here the liability, and the liability is just the combination of genetic and environmental factors. So we have a distribution, and everyone in the population will sit on this distribution somewhere. Might be here, might be here, might be here, might be here. Okay, so in this very simplistic model, we're not considering the sex bias just yet. We can say that if we meet a certain threshold, which we would call tau, we have enough genetic and environmental factors that we will develop the phenotype. In this case, it would be autism. And if we are over here, we won't have enough. So you can basically say that unaffected individuals are here and affected individuals are here. And that's a very simplistic way to think about it. But when we introduce the sex bias, we have to think about it just a tiny bit differently. And the way that we think about it differently is that we consider that males would have a threshold that's a little bit lower than the female threshold. And so what that would look like is we would have unaffected males over here, and we have affected males over here. Likewise, we have unaffected females here and affected females here. And so what that means is there's actually one little tiny bit of this distribution where we have females who would not have autism and males who do. Okay, so I've shown you this kind of theoretical model, but what could actually be the reasons underlying this? And I like to think of them in terms of an acronym that I call SHIP, S-H-I-P, and this is one of my favorite ships actually, the Lady Washington. I did some of my training in Seattle, Washington, so it's a really cool ship that you can ride out there. But what it stands for in the context of today's talk is the S stands for sex chromosomes. So you may remember that as females have 2 X chromosomes, whereas males have 1 X chromosome. Males have a Y chromosome and females do not. There are other considerations like some X chromosome genes can escape X inactivation, and we'll talk more about what that means a little bit later. The H stands for hormones like estrogen and androgen. The I would be for imprinting, so this is some kind of a sex-specific imprinting that could be occurring. And the final is called the protective effect, which in some sense can sort of encompass the 3 above it. But the protective effect says that a female would need a greater load of mutation, either in terms of severity or in terms of number to become affected than would a male. And you've probably heard of this as something called the female protective effect. And so a while ago, I thought about, I've been thinking about this for some time, but I thought about what is the evidence that we have that this female protective effect might actually occur in autism, as well as in other neurodevelopmental disorders. And so that's what I'm going to show you next. So actually, if you look over the course of the years from 2011, even to this past year, 2019, there have actually been a lot of findings that support this idea of a female protective effect. And probably one of the earliest published results was from Levi in 2011, where they were able to show that there are a higher frequency of de novo copy number variants in females with autism. In 2012, Neal et al. Were able to show an increased rate of de novo variants in females with autism. In 2013, Robinson actually was able to show that there are more autistic impairments in siblings of females with autism than males with autism. In 2014, Jacquemont was actually able to show that there's an excess of deleterious autosomal CNVs in females with neurodevelopmental disorders more broadly, but also including autism. In 2015, we were able to actually show that there's a greater gene discovery rate when we look at female-enriched multiplex families, where we could find novel genes by focusing on families with 2 or more affected girls. In 2015, Sanders was able to replicate the effects that Levi saw in 2011, where you have these higher frequency of de novo CNVs in females with autism. And then in a really beautiful paper in the last year, Doan et al. Were able to show an excess of females with recessive gene disruptions. And this really added a new kind of paradigm to what we had already known in a new area of space for this female protective effect. So this is all quite consistent with an idea that more severe mutations, in particular what we call rare variants of large effect, are seen in females to develop autism. But what I wanted to point out is that all of this work has very much focused on a pooled approach, where we have both males and females being represented in the study. And as a geneticist, the reason why we would do this is because we need really big sample sizes to have power to detect significant genes, and that's a really important consideration. But we and others had hypothesized that if we had actually focused on females with neurodevelopmental disorders, maybe we could have had a greater gene discovery. And that kind of led into this new study, which I'm going to talk about now. So the three main questions that we asked in this new study were, are there any genes that are significant in only one sex and not the other? The second question was, is there a differential gene discovery rate in males and females? So that really just means, is our gene discovery really greater if we looked at females only, as the female protective effect has suggested? And then finally, are there genes with differential counts of de novo variants when we directly compare males and females? And to our knowledge, this had not been done before, so it's really quite an interesting area to work in. So this is the framework that we developed. As I mentioned, our paper was published in the American Journal of Human Genetics late last year. And so I would, if you want to check that out, just let me know if you need a copy or anything. Be happy to send it to you. But what we did, what I want to first talk about is kind of the standard approach of pooling the samples together and how we would ask statistically if a gene is enriched in autism. Okay, so what we did originally in all these old published studies is to combine males and females together. And then we used two different statistical models. And I do want to point out there are other models that you could use, but these are two that are pretty well established. And these are called the chimpanzee human model, which was originally created by Brian O'Rourke and was recently updated by Bradley Coe. And this is a model which asks, are the number of mutations that I see in a given gene more than I would have expected by chance based on the difference between chimpanzee and human? The other model is called de novolizer, and this was developed by Caitlin Samoka. It's a really cool test that actually looks at the sequence content and asks, based on the sequence content in that gene, are the number of mutations that we see statistically significant? And so what we did was we did not create a new test for this paper. We actually said, let's run these same tests on either males only or females only. And that was our first approach. The second approach was to say, let's compare males directly to females, and we can use a very standard statistical test for this called the Fisher's exact test. So as I mentioned, having a large number of samples is quite important. And so over the years, we have been developing a database called de novo DB. And this database's goal is to collect all of the variants from the literature that are published and put them into a database where other researchers can go and look and begin to ask questions that might be of interest to them. And so what we got from the database was almost 9,000 sequenced parent-child trios. This represents 26,475 individuals, so it's a very large sample size. And we had de novo variants from the version 1.5 of the database and a sex ratio of 2.2. So we included both autism, intellectual disability, and developmental disorder as one large cohort in this analysis, which we called neurodevelopmental disorders, or I will continue to refer to it as NDDs on my slides. So the first thing that I want you to see is the genes that we actually identified as significant in females or males. And so let me just walk you through this slide. So first of all, on the bottom here, I'm showing the level at which we would have exome-wide significance, and that's at a level of 4.3 times 10 to the minus 7. That's just the p-value cutoff. It's actually a very stringent statistical correction. And what we found were there were 36 genes significant in females, Those are shown in this yellow circle here. There were 37 genes significant in males, and that's shown in this blue circle here. And there are 54 in the whole set, so when you add these all together. So what you can see is that there are actually some genes in the middle that when we look at females and males separately, right? Some of the genes are significant in both, even though we're looking at them separately. An important consideration before I get too far is that we actually do not have good gene models for the Y chromosome for these particular tests, at least when we ran them. And so that is sort of an important consideration given the importance of the Y chromosome. But today we're just focusing on the autosomes and the X chromosome. So what I also did here was put this dotted line in. And above the dotted line are all the genes which are on the X chromosome. And below the dotted line are all the genes which are on the autosome. So that would be chromosome 1 to 22. Okay, so you can see that our gene discovery rate, if you just looked at the raw numbers, they look very similar in males and females. But what I want to point out is that we had only about 3,000 females and we had about 6,000 males. So that means that actually our gene discovery rate, just by looking at this, was greater in females because we had half the sample size and we still discovered the same amount of genes. Okay, so that's just one way to look at it. But I think statistically we would want to say, you know, can we empirically ask if that truly is significant? So what we did was we created this test called a downsampling analysis. And this is a common test that people will use to try to ask these kind of questions. So we knew that we had 2,779 females. And when we studied those females, we found 36 significant genes. So the way the downsampling works is we will take our big set of males, right, 6,000 males, and we will randomly pick 2,779 of those males out. And we will do the test on those different samplings. And in this particular paper, we did this 10,000 different times. And so what this looks like is this. You'll have your actual female discovery rate, which we had 36 genes here. So this is the number of significant genes in those tests, the two models. And here are the number of downsampling tests that we did. So this is what we're looking at, right? We want to say, when we look at only affected males, do we ever get close to this number? And when we did these 10,000 tests, you can see that our gene discovery rate was never at this level. It was always lower. And that was significant. So that suggested to us that our discovery rate was actually statistically greater in females in this context. We also added an assessment where we looked at the different sets, but we considered males and females together. So this means that when we got to 2,779 people, they could have males or females in there. And as you might expect, the distribution did shift up. It was still significantly more in the female only, but you can begin to see that we can start to reach that level of significant genes when we begin to add females back into the analysis. Okay, so this was really intriguing to us. It seemed to go with the female protective effect, and we were very curious as to why this could be. And could there be some kind of neurophenotype driving this effect? And so the thing that we thought to look at was the correlation between individuals who had a developmental disorder and individuals who were female in any particular set where we had males and females together. And so what I'm showing you here on the x-axis are the fraction of female individuals in a particular random sampling set, and the fraction of individuals that had a developmental disorder in that particular set. So for here, you could say that about between 0.30 and 0.31 fraction of individuals were female, and maybe like 0.5 of them also had a developmental disorder. But what you could see was that there's this correlation between them. It is significant. That suggests that perhaps the number of individuals with a developmental disorder could be driving this effect. And we also looked at individuals who had autism and had IQ data available. And for this, we were able to show that the female had lower IQ than the males in this particular analysis. And this is very consistent with published results by Iosifov and others in the past. So this could be the reason why our gene discovery rate for these rare variants of large effect was greater in females. Okay, but I now want to go to the genes themselves. And I mentioned this discovery cohort that we got from the de novo DB database. But what I also wanted to mention is that we did something that I think is quite different than what most people have been doing for de novo variant analysis. And that is to seek a replication cohort. This is not uncommon in other types of genetic studies, but it is fairly uncommon in this. And what it does is it gives us a better statistical stringency to say whether a gene is involved or not. And so we paired up with some people at GeneDx, and we were able to look at 18,778 additional sequence parent-child trios. So this represented another 56,000 individuals. And GeneDx was very kind to give us de novo count data for each of the genes that we found significant in this previous work. And the sex ratio in this particular cohort was 1.2. So this is the same type of graph that I showed you earlier, where we have the females shown in this kind of yellow color and the blue shown in this, the males shown in this kind of blue color. Again, we have the X chromosome genes here and the autosomes here. We saw that there were 22 genes significant in females, 18 in males, and 25 in the total set. But again, this shows that the gene discovery seems to be greater even after replication when we look at females. So I thought I would show you more than just a graph like that. I'm going to show you some real numbers and how we would look at it as geneticists. So this is something that we often call a Manhattan plot. And you may have seen them before, but Manhattan plots have chromosome on the x-axis. So this would be chromosome 1, 2, 3, and going on out to the X chromosome. And then we have this thing called the minus log 10 p-value. And all you have to understand about that is that the higher the dot is, the more significant that particular gene is. And what I'm showing you in red on the left are the genes significant in females, and in blue, the genes significant in males. Now, you'll notice there are some other dots here that don't have labels, but that's because they were significant in both males and females. And so we just didn't label them because we wanted to focus on the genes that were, female specific, quote unquote, genes, or male specific, quote unquote, genes. But we really wanted to be sure, like if you're gonna say a gene is really seen only in females or only seen in males, you gotta be really sure that that's true. And so what we did was to take these genes, and now I'm showing you a table which corresponds to these graphs, where we had genes which are significant in females, shown here, and significant in males, shown here. This is the chromosome that the gene is on. This is the minimum p-value in our statistical tests in males and females in that de novo DB discover cohort, and then in males and females in the replication cohort. So now I'm just shading in all of the ones that meet the most stringent statistical correction that we can do, which we call multiple test correction significant. And as you can see, the female ones are significant in females, and the male ones are significant in males. That's exactly what you would expect. So we thought, okay, there are different levels of statistical significance we can assign. And one level is to say it's nominally significant. And so nominally significant could be suggestive that the gene could be important in that particular test, right? I mean, the multiple test correction significance is usually what people consider as the gold standard. But we thought, what if we just looked in the other sex? So for females, we'll look in males, and for males, we'll look in females. And so what we saw was in gray, there are some genes, like all of the genes that are significant in only males, reach nominal significance in females. Now, that's suggestive that if we had more individuals, they might have become significant in females. And so we would probably not say these are male-only genes. Likewise, there are a few genes in females like that, where ITPR1 is also looking significant in males. So there are actually just 4 genes which truly look like they're significant in only one sex. And those are genes that are significant in only females. They are DDX3X, HDAC8, NAA10, and USP9X. And they all reside on the X chromosome. And remember that females have 2 X's and males have 1. And so I want to tell you a little bit more about this finding, because after we finished the study, we thought, wow, that wasn't that surprising. But at the time, we were a bit surprised. And so what we saw, if we just take all of the genes that are on the X chromosome, I'm actually including all 5, but we can see that this is what we call in genomics an ideogram. This is just a representation of the X chromosome, and these bands are patterns that we would see. But this is where the genes are actually residing on that chromosome. And what was really interesting to us, that was 3 of the 5 genes that were significant in females were either constitutive or facultative escapers of X inactivation. And this is not complicated. I'm just going to talk you through it real quick. So in females, we have 2 X chromosomes. But most of the time, one of our X chromosomes is silenced. So we express genes from mostly just one of our X chromosomes. But 13% of X chromosome genes are known to escape this inactivation, which means that they, in females, are expressed from both copies of the X chromosome. And so I'm showing you expression data for this gene called DDX3X in males and females. And I took this data from the GTEx consortium. It's a very wonderful data resource. And on the x-axis are the different tissues. So I picked some tissues in the brain, the heart, and the liver. This is just to show a representation. And this y-axis is a metric of expression. And what you can see is that females do indeed express more of this gene than males. And that's very consistent across different tissues, as you can see. So this made us really think, okay, so this is consistent with a model where females have a buffered expression and they can develop the neurodevelopmental disorder, whereas a male with that same mutation might not, survive. So that may be why we never see, or almost never see, males with mutations in these genes, because there is this buffered expression. But going back to that idea of Fragile X syndrome, it's different. It's really different than Fragile X syndrome, because in Fragile X syndrome, females more often do not have the syndrome, even though they have the variant, whereas males do. So this is another way that we can think about the X chromosome's role in neurodevelopmental disorders. And it's an area that needs further investigation for sure. But there is, really some great news about all of this, is that over the years, clinicians, while we have statistically shown this, and I mean, these genes are very significant, we know that, but clinicians over the years have worked to collect patients with variants in these genes, and they have been able to give us very important insights into what the phenotypes are. So for example, USP9X and DD3X have been shown to be involved in intellectual disability in females. WDR45 is in neurodegeneration with brain iron accumulation, which mostly affects females. HDAC8 is involved in Cornelia de Lange syndrome, with more severe intellectual disability in males than females. And NAA10 is also known and is involved in Ogden syndrome, which actually affects both males and females. So this is amazing to see how the work that we have done over the years is really contributing to our understanding. So the final analysis that we did was to do this comparison between males and females directly. And the reason we did this was because we actually thought there would be quite a few genes that would have a really big difference between males and females. And I guess the, the message that came out of it was actually there's just one. And that one gene is called DDX3X. And this gene in our discovery cohort had no variance in males, but it had 26 in females. In our follow-up study, there were actually 2 males who had variants in the gene and 65 females. So this was very, very significant. Now, I just wanted to point out that the Share and Silver Labs have just put out this beautiful paper on DDX3X and the function and phenotype follow-up of variation in this gene. It's just a beautiful study, and I would refer you to that. But we thought, boy, we had so many samples, tens of thousands of samples. What would be the next set of genes that might be significant if we had even more? And so we just said, what if we go back to that idea of nominal significance? And it turns out there were 3 genes that had nominal significance in the discovery and replication, and they were, again, all on the X chromosome. So MECP2, NAA10, and HDAC8. Okay. So we thought there might be a few more genes, but how many more families would we need to study? And this is where things like the SPARK cohort are really leading the way. And so we did this thing that we call a power test. And a power test is a way, can be done many ways, but it's a way to say, based on the data that I have, what would I need in the future to see more significant genes? And so it's shown in a table view and in a figure view here. But essentially, if we added 10,000 more trios, we would still have just one significant gene in this comparison. And in fact, we have to get out to 50,000 more trios to see 3 new genes. And those will likely be the genes I showed in the last slide. However, once we get out to much bigger sample sizes, sure, we're going to see a lot of differences between males and females, but these are probably going to be very small effect sizes. And so I think the take-home message here is that there are actually a few genes, in particular DDX3X, that seem to be really different in terms of these rare variants of large effect. So I'll just conclude this part, and then I'll get into the last part, which is pretty short. So I just wanted to say that sex bias is an important consideration when we assess a phenotype. We do need to understand why this happens from genetic and non-genetic perspective. I wanted to show you that gene discovery based on rare variants of large effect is really greater when we look at females with neurodevelopmental disorders, and it can reveal important insights about the X chromosome. However, sorry, I'll get back to that in a minute. But while studies of these approaches have used combined pool approaches, if we were to do other phenotypes in the future, we might think more about studying these rarer classes. But probably we don't need to do that for neurodevelopmental disorders too much. Okay, so I want to now talk a little bit about going from the genes to the biology. But this is a very important area of our work. I think there are 2 main things that we really have to, from my perspective, that we have to do on the genetic side of things. And one is we really need to understand the genes that are involved in autism and other neurodevelopmental disorders. We need to know what those genes are. And that's where things like the SPARK project are really exemplars, and they are leading the way. And it's a very exciting area of investigation and very important. The other is to try to understand more about the genes that we already know are important. And that's where things like that DDX3X study I mentioned, where they dove into the function of that gene and the phenotype in patients, is really important. Okay, so this last little bit is about gene regulatory networks in autism. And so this is an idea that's been around for a while and primarily in the development field. But it's this idea of how we can model these biological interactions. And so gene regulatory networks, they are composed of a set of things we call regulators that work together to control output of RNA and protein levels. So this is stuff going on in the cells. So we have some kind of input into these networks that directly would affect the molecular output, so RNA and protein, and ultimately would alter cell behavior and function. So let me just introduce you to an example of what this would look like. So these gene regulatory networks use things that we call cis-regulatory elements that regulate multiple genes. And these genes can regulate other genes. So this idea is that not all genes are doing the same thing, right? That's a pretty understandable thing to say. But we have to sort of understand where they fit. So if we had a gene, which is kind of represented by these little lines here, and we said this is gene A, this gene A actually can affect lots of other genes down here. Whereas this gene B doesn't really have much downstream effects. So let's just model this. So if we were to remove gene A from this regulatory network, that would be very bad. It would be rate limiting to the network because now all of the downstream things can't happen. Whereas if we delete the B gene, that would only have local effects. And so we just really thought a lot about this, like what could this mean in terms of the sex bias? And I think one thing that we thought it could mean is that maybe females are having mutations in these genes that are more rate limiting in the network as compared to these B genes that we may see in males, excuse me. So it's just one thought. So then the idea would be that the genes we saw significant in females might be regulators of other genes. And so we revisited this significant gene set, which if you actually look at it and you consider that these genes were nominally significant in females, you could say that our gene discovery really was mostly in females. And so we could take this set of genes and ask biologically, what are they doing? And the first thing that we looked at was the expression, and their expression in these things called inhibitory neurons in the cortex and excitatory neurons in the cortex, and then in these other cells which are not neuronal. And what we saw was that in these dotted lines are all of our significant genes. And so these lower lines indicate that these genes are significantly enriched in more cell types in the cortex than some random control genes. And because these lines are more separate from one another here than here, that this is actually more significant in neuronal cell types than non-neuronal cell types. So that's just telling us that these genes are probably important in neurons in the cortex. What I thought was really kind of more interesting was to ask how they might be fitting together. And so of the 25 genes that we had, remember they all code for proteins, 22 of them actually sat in a network together. And this is called a protein-protein interaction network. The size of the bubble corresponds with the number of variants that we saw in that protein, in the gene that codes for that protein. And the darker red it is, the more that we saw female cases than male. And the in the darker blue, the more male cases that we saw. These proteins are associated with transcriptional processes and chromatin binding. And if you've been following the genetic work on autism, you would know that this is one of the main biological processes that's been shown over and over as important. The other one is synaptic function, but it's very interesting that these proteins in particular are these kind of chromatin binding genes. And as you might expect with that role, they mostly localize to what's called the nucleus within the cells, and that's the area where this chromatin kind of binding work would be going on. And the important thing to note here is that many of these proteins then, because of that, are key regulators of other genes. And so I think that's kind of an interesting result from this work. So I'll just briefly conclude this in saying that these gene regulatory networks are really important for us to move from our gene discovery to actual molecular function. We can study these rarer individuals to try to learn the genes that might be rate limiting. These genes are very connected to one another and are responsible for regulating other genes. Okay, so I'm just going to do my final conclusions now and just say that sex bias is really an important consideration when we are assessing phenotypes. And it's good to see that the field is, oh, I'm sorry, that's my phone, is really working on this area. Sorry, I'm at my house, I'm sorry, in my apartment. So gene discovery based on rare variants of large effect is really greater in the rarer class and can reveal important genes on the X chromosome. Oh boy, 1 second, I'm so sorry. Okay. I'm sorry. Okay, so however, it's important to note that most of the significant genes that we have discovered are not exclusively discovered in males or females. So while the rate of discovery is different, the genes really aren't that different. And so I think that's something we need to wrap our minds around and understand. So work remains both in the genetic side of things. So one thing is we don't understand the possible role of the Y chromosome yet, and we don't really understand the inherited variation possible contribution to the sex bias, both in the forms of common and rare variants. And of course, as you would probably know, there are many non-genetic factors that we still have to understand as well. So I will stop there and take any questions, and thank you for your time today.
Thank you so much. We did get a few questions in. If anyone has any others, feel free to put them in the chat. The first one we have here is asking about if you've used genetic data from Spark in any of your studies. You may have mentioned it, I can't remember.
Okay, so in our published studies, we have not, but I think that's an area that's really important. So this new set is being led by the Spark Genomics Consortium, which I'm a part of, really working on this new set of data that's coming online. And so that is an area of future investigation, that's for sure. It's not included in the work that I showed today, though.
Okay, all right. And then at one point you had mentioned estrogen and progesterone in your SHIP model, and someone was curious about if testosterone would also be of interest.
Oh, yes, of course. That would also be an area of considerable interest. I just wrote a couple of them as examples. I think that's an important area in the investigation. Yes, definitely.
Okay, and here's another one. Do we know if inherited variants are substantially different than rare deleterious variants?
Well, I'm not quite sure I understand how the question is being framed, but there are differences between inherited variants. Just saying inherited variants, right? You could be considering variants that are common and present in lots of people in the population versus those which are really rare and maybe exclusive to a single family. And certainly the genetic signals in those two contexts are very different. I'm not sure.
Okay. Yeah, someone was clarifying inherited versus de novo, if that helps.
Oh, inherited versus de novo.
Yeah.
Yeah, so that's an area that's actually, we have a, from my postdoc lab, actually Amy Wilfert, whose name's on the slide there, she just led up a study that's on bioRxiv right now, which is talking about the contribution of inherited variants in terms of autism. And I would reference you to that. So bioRxiv is an open access, so you can download the paper from there. But showing that, yeah, there are some differences between the kind of biology in inherited variation versus de novo. Yes, that is true.
Okay. So does any of your work suggest that females with autism would be more likely to have genetic finding than males? That's kind of a big one.
Right. So based on all of the female protective work that's been done over the years, it certainly does suggest that a female would be more likely to have a rare variant of large effect than a male. But, you know, there are some things we have to consider when we think about that. So that's why we did that phenotype test to say, when we look at those individuals, is it maybe a difference in the ascertainment of those samples in terms of whether they have intellectual disability or something else? And I think it's a bit challenging for us to untangle those two observations, but I would generally say probably yes, with the caveats that it's a little more complicated.
Mm-hmm. Okay. At one point you had showed a slide with IQ data. Someone was wondering if you could go back to that.
Sure, I can try. Let's see here.
I think they just wanted to see what the differences were.
Yeah. Oops, that one. I think that's the only one I had maybe on IQ.
Oh, okay, yeah, the female mean and male mean.
Mm-hmm. Yeah, and that is a very, very concordant with what Iosifov found. I think it was in 2014 paper. Yeah.
I don't know. This one might be a little complicated, but
Okay.
Do genes changed by chromatin remodeling have anything in common?
Well,
Broad question.
I guess that is a broad question. So, I mean, they are chromatin regulators. Many of them have what you would call like domains, like similar types of domains within them, if that's kind of the question. The patterns of mutation can vary though. Some have more clustered mutations than others. But yeah, biologically, because they are doing similar things, they will have some similarity to one another. I think hopefully that kind of answers that a little bit.
Okay. In your studies, have you seen any differences in where the mutations fall in given genes between males and females?
Oh, no. Okay, so I was shaking my head yes first because generally we have found genes where there are clustered missense mutations. One that I can think of is like KCNQ2, CTCF, but we have not done that test yet in males and females. One reason is because I developed a test that can do this, and we did this generally, but we are actually working on a method to vastly speed up the rate of doing that test. And so as soon as that is ready, we will run that on the males and females separately. So that question has not been asked yet of the data, but will be in the next few months.
Okay. Do you have concerns that our diagnostic tools for autism may be better suited to identify autism symptoms in boys, but not in girls? And does this have any impact on genetic research, given that, I guess?
Right. So I first want to point out that I'm not a physician. I am a research scientist, so I don't actually see patients in the clinic. So I just wanted to make that very clear. But I do think that that could be an important aspect of things, right? So depending on how the samples are, what we call ascertained, could really affect the genetics that we do. And I think that's something that, if I could just say for a moment, is really interesting about the SPARK project itself. Because the SPARK project is much more of a kind of open enrollment, like you can enroll from anywhere. And I think that's really interesting. And so one thing that we found in the very first SPARK paper that we did was that a lot of the genetic results that we saw in that analysis were very similar to what we had seen in cohorts like the Simon Simplex collection, where the data was much more clinically oriented, like much deeper clinical workups. And so that suggests that maybe that's not as big of a thing as we thought it would be, but I still do think that's a very important consideration. Yes, and we do think about that.
Okay. Um, let's see, I'm not seeing any other new ones coming in. There were some questions that were pretty specific. So
Okay.
if people have specific questions about, like chromosome changes or
Mhm.
gene changes, we recommend that you email us at info@sparkforautism.org, and maybe one of our geneticists on the team can help to get an answer. If